So the structure of DNA, we have Watson and Crick, along with Rosalind Franklin down here, and Wilkins and Pauling, that they developed a double helix model of DNA. Other leading scientists worked on the question, such as these people that we just mentioned. This was a pretty exciting time for science in the 50s. If you're interested in some good science, historical reading, track some of these guys down. It's definitely an exciting time in the science field. So, again, here's Watson and Crick, the guys that are credited. I have an Englishman and an American that were both credited for working together with figuring out how DNA is actually structured. So, Rosalind Franklin is the one who came across that it's actually going to, this image here, that it's actually a, how they're base paired across, and that they have these rungs. Didn't quite know that it was double helix. So, but how is DNA copied? So it can be a couple ways. The replication of DNA, base pairing suggests that it would allow each slide to serve as a template for a new strand. Here's the quote from Watson and Crick. "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So, there's three potential models of DNA replication. We have one that's conservative, meaning that these two blue lines always will come back together. These are all the same strain, meaning that you will end up, this one will come down and create two identical ones. Here's semi-conservative, meaning that one strand will form as a template for another, and so ultimately you have two strands, so one double-stranded DNA will form two down here, and then two will form four. Again, that's called semi-conservative. The dispersive is just kind of random sections throughout. So let's go ahead and look at this. Let's design an experiment to verify this. So let's see. This will be the parental. This is the first generation, and then this will be the second round of replication. So let's look at this. Semi-conservative replication. This is Meselson and Stahl. They label the parent nucleotides in the DNA strands with heavy nitrogen, okay, nitrogen 15. And they label the new nucleotides with the lighter isotope, nitrogen 14. So go ahead and stop and make some predictions. If you need to rewind to see what it is, what the three different possibilities, you may do that. For the sake of time, I'm going to move forward. So after he labels it, we have, here's the parent that's again labeled with 15. And here is the second replication that is labeled with 14. And so we're going to take DNA after the first replication, and then we will take DNA after the second replication. So here is the 15 parent strands. Will look like this. We'll get a band when we look at it like that. And so let's go ahead and make our predictions here. So here's the parent strands. So we have 15, and we're using this to be red. So the conservative, that's how it looks like. And then also the 14 will look like this. Again, this is after the first round of replication. So this is what we would expect. This is a prediction. So the semi-conservative, you would have a mixture of the two. So you will have half of the DNA strands will be have one that was blue, and the other half will one that was red, or one that was 14 and one that was 15. So they're represented here with both 15 and 14 and a purple color. So dispersive will be the same but a little bit different. It isn't one strand, it's just kind of randomly mixed. So let's go ahead and look at the second round of replication. For the conservative, we would expect to see one that's red, and then a new one that's all blue, or 14. For the semi-conservative, we would expect to see another purple or mixture of 15 and 14, as well as one that's just 14 and 14. So we'll have a blue band. And the dispersive, we would end up still, again, because it's just kind of randomly mixed throughout, we will end up with one that's 15 and one that's 14. So let's see what actually happened. here are the strands. So this is segments of DNA that have been drawn out on an electrophoresis. We'll also do a similar experiment to this, at least where you get to see gel electrophoresis. So here's the parent. Okay, here's the first replication. And this, so from here we can determine that we end up with all sort of 14 and 15. So we know that this has not happened. So at this point, after the first replication, it is either semi-conservative or dispersive. So we have to see the second replication to see which one is correct. So we can now see, we know it can't be conservative, and it also cannot be dispersive, because we do have a band here that represents 14 only, not mixed with any 15. Therefore, semi-conservative is how DNA is replicated. So let's summarize this here. The march to understanding the DNA is the genetic material. Morgan said that genes are on chromosomes. Griffith said that a transforming factor can change the phenotype. Avery McCarthy McLeod said that transforming factor is DNA. Chargaff came up with Chargaff rules that A is equal to T, or adenine is equal to thymine, cytosine is equal to guanine. Hersey and Chase said that confirmation that DNA is the genetic material. And Watson and Crick said determine that the double helix structure of DNA. And Meselson and Stahl came up with a semi-conservative replication. So the central dogma, the flow of genetic information in a cell, is from DNA, replicates itself by creating new DNA, and then DNA can be converted into RNA through the process of transcription, and then RNA gets converted into protein through the process of translation. So the rest of chapter 16 will be devoted to replication, and then 17 will be devoted to transcription and to translation. So please come to class with any questions. This is a photo of a lot of these famous scientists together at a party that were just mentioned. So a lot of these guys hung out together at Harvard. So thank you.